Great honor to be here, and so I have a little story I'll tell you about Irwin in a second, but I also wanted to reinforce, Professor Montgomery, so I also wanted to reinforce the importance that Fred Haer has had on neuroscience and the field of deep brain stimulation in general. It's really a testament to the goal of every engineer in the world is to build something that other people will use and then get benefit out of. And almost all the kinds of neurophysiology data that I'll talk about today was for the most part probably almost de facto collected using FHC electrode technologies. So sometimes we lose sight of those things in our day-to-day activities, but it's these fundamental pieces of technology that allow us to do the things that we want to do. And then a little tidbit about Dr. Montgomery. So when I was a graduate student, I was working on some questions related to the basic biophysics of how electric fields interact with the brain. And it was, these were interesting and fun mathematical questions and neuroscience questions, but I didn't really necessarily have a goal that I could take that knowledge and apply it to. And so as a young student, I attended a seminar that was given actually by Dr. Montgomery about deep brain stimulation, this brand new thing that had just kind of come on to the medical scene in the United States. And that was literally a life-changing moment in my life. So I saw what Dr. Montgomery was doing, the amazing impact you could have on these patients, and this burgeoning sort of new technology bed, if you will, for interfacing with the nervous system. And it was so completely obvious to me then at that moment that this is what I could do with my life that would actually be useful and productive. And so, so I always will have that debt of gratitude towards Dr. Montgomery for opening up my mind, if you will. And so it's so exciting to now hear about him teaching in this sort of liberal arts environment and bringing a new perspective to this concept of medical ethics and whatnot. So really, really fun and exciting times. Okay, so now on with the talk. So I typically talk to neurologists, neurosurgeons, and systems level neuroscientists, and typically, it's a room full of other people that are experts in the same things that I do. And so when presented with the challenge of talking to a much broader range of people, it's kind of hard, actually. One of the more difficult things to do as a scientist in trying to find a way to put together a talk that other people can appreciate and understand. So if it isn't working, just let me know, and I can try and adapt. So feel free to, if I say things, say jargon or abbreviations that you don't understand, just raise your hand and say, what does that mean? I'm happy to try and adapt. So anyway, with that little caveat, I'm going to try and talk a little bit about how we've been using basic neuroscience to then also provide some kind of indication or understanding about what we might be doing with deep brain stimulation technology. So deep brain stimulation is a medical therapy that is relatively widely used now. Almost 100,000 or over 100,000 human patients have been implanted with deep brain stimulation systems around the world, primarily for the treatment of Parkinson's disease, but also for all kinds of other movement disorders, things like dystonia, essential tremor, and now starting to branch into other neurological disorders like epilepsy and neuropsychiatric disorders like depression and obsessive compulsive disorder. So we have a wide range of different therapies that are either under development or already have approval from the FDA. But all of them reside or work with these basic fundamental pieces. So we have our implanted pulse generator, and then we have our permanently implanted deep brain stimulation electrode that would then be targeted to specific brain regions. And I'm going to try and talk to you about the process of how that targeting goes forward, and why they would select a given target, and then some of the, let's say, complicating scientific factors that we try and understand once those electrodes are in that target. So, but before I get into that, I just want to reinforce sort of the excitement, if you will, on Wall Street, and hopefully someday on more wide scale in the development of this neuromodulation market. So, as a historical footnote, neuromodulation as a field, as a clinical application in human use, really got its start at Case Western Reserve University. Well, at the time it was Case Institute of Technology and Western Reserve University Hospitals. And in the late 60s, an engineer named Tom Mortimer and a surgeon named Shealy got together to create the first spinal cord stimulation system for the treatment of chronic pain in humans. And spinal cord stimulation is now actually a gigantic market, a multi-billion-dollar a year market that takes up about half of this total field of neuromodulation. The next biggest piece of that whole neuromodulation world is deep brain stimulation. And then there are a whole host of other things that are sort of new fields in the world of neuromodulation. So electrical stimulation to control the nervous system and then have subsequent action on other organs within the body. So gastric, bowel and bladder function, all kinds of different activities. So basically everything that your body is capable of is innervated by or led into by the nervous system. And if you can then tap into the activity of that nervous system with an electrode or with electrical stimulation, you can override or overcome any deficits that might be in place because of some kind of neurological disorder or disease. So with all that kind of opportunity out there, it's no surprise that this market is growing quite strongly. So right now it's about a $3 billion a year market, but the projections are for really substantial growth in this area. So there are great financial opportunities, and then more importantly, there are great therapeutic opportunities for us to take these foundational pieces of technology and put them into place for use on a much broader scale. And so as an engineer, that's I look at as my goal. What do we need to build these platform technologies so that they can be useful on a much broader scale? So this is what the technology looks like today. So this is an example system from Medtronic. There are also systems available in different parts of the world from Boston Scientific and from St. Jude. So these big multinational conglomerate companies that build medical devices. But all the systems look, for the most part, the same. And so with this particular system, the Medtronic system, you have your implanted pulse generator that's connected by wires to a connector and then to the actual stimulating electrode contacts. And so in this particular model, there are 4 individual electrode contacts that could be stimulated through. And then this is the piece that's implanted in the body, or in the brain and in the chest. And then you've got this blue thing. This is the clinician programmer. So this is the device that the neurologist uses to connect with and talk with the implanted device so that they can have some kind of control over the stimulation parameter settings. And then this black device is the controller that the patient has. And so they can take this home and they can turn the device on and off with it and things like that. Okay. So the process of actually having a deep brain stimulation surgery is a daunting one. So if you have reached that point so most of what I'll talk about today is deep brain stimulation for Parkinson's disease, because that's what I work in for the most part. And so there are many really effective medications for the treatment of Parkinson's disease. And so for the most part, you can have that disease and live relatively well for many years after you've been diagnosed. But at some point, most patients will develop things like drug-induced dyskinesia. So the drugs themselves actually start to cause a number of side effects. And the progression in the disease gets to the point where you might need something else to help you improve your quality of life. And that's where you might turn to a surgical therapy like deep brain stimulation. And so the goal then would be to, obviously provide improvements in your motor symptoms, and that would then obviously have some kind of improvement in your overall quality of life. So, but it's a big decision to make because you have to undergo, an invasive brain surgery to have this technology. And so this is sort of a picture of how that process would look. So the day of the surgery, you'd have this frame placed on your head, and actually FHC actually makes an innovative technology that I'm not talking about right now that are actually these platforms that you can do that replaces this permanent frame. But the traditional surgery uses a frame like this. And then you've got this arc that they can use to then have a projectile or a microelectrode and electrode delivery system so that they can have a pinpointed stereotactic reference frame for them to place their DBS electrode. And that is augmented by surgical software technologies. This is an example from Medtronic of their what's called the Stealth Station. There are many different companies that make these kinds of surgical navigation software technologies. And so, but the goal is that you need a stereotactic coordinate system, a fixed coordinate system relative to the patient's head, and that's what these types of frame systems provide you. And then you want to have your imaging data, so obviously MRI imaging data, that can help you identify structures in the brain that you would want to put your deep brain stimulation electrode. And so if you can see where you want to put that electrode on the image, and you have this fixed coordinate system, then you can dial in the parameters of your frame so that you will have a trajectory that will take you to that place where you want to be, so then you can put your permanent electrode. So once that electrode is in the brain, then the next step of that process is what they call DBS programming. So now this is the job of the neurologist to take what the surgeon has given them in terms of the placement of the electrode and customize the stimulation so that you maximize therapeutic benefit and minimize the side effects that would be generated from this electrical stimulation. And this process goes through a typical scenario of doing what they call monopolar review. So as I told you before, there are these 4 electrode contacts, and they would turn on these individual contacts such that they would test whether or not they can get therapeutic effect through each contact, and they would do that sequentially as you go through the system, and then try and find the contact that would have the best, what they call therapeutic window. But this is a very time-consuming process, many hours. The quantitative estimates that are available in the literature would suggest that this process takes about 8 clinical hours of time in terms of balancing and selecting this appropriate stimulation parameter settings, and then also subsequently balancing their medication dosages. So when I first got into this field, that was really the first thing that I thought, okay, this is a problem that we can come up with an engineering solution to address. And so the concept that I really wanted to try and approach was a concept of what we call patient-specific deep brain stimulation models. And that the idea was that if we could take that imaging data that was already being collected for the surgery, if we could then take that imaging data and that kind of anatomical knowledge and then put it into the hands of the neurologists, it would provide a new platform for them to speed up that DBS programming process. And so if we could then provide them with some kind of visualization, they would be able to, instead of spending 6 to 8 hours, they could do that customization process in a much shorter amount of time. And so, but we had to come up with some mathematical techniques that would allow us to have some kind of a visual reference for the spread of stimulation, right? So if you have this electrode, you're using electricity to modulate the nervous system, there is a dose that you are injecting, right? It's kind of like a drug because you have a dose of electricity as opposed to a dose of a drug. But there wasn't at the time a way to quantify what that dose really would be. So the concept that my group really tried to develop was calculating using computer models, and I'll try and explain a little bit about what those kinds of models are, of a volume of stimulation and how that volume of stimulation would change as you change your stimulation parameter settings. And then, how would that interact or overlap with the brain anatomy and the individual anatomical structures that we're placing our deep brain stimulation electrodes into. So there are sort of a series of what I would consider fundamental or basic questions that we would need to really get at if we were ever going to get to that point of having some technology that could, take science and put it into the hands of a neurologist to then subsequently actually have a clinical effect. So the first step in all of this is, what is the electric field generated by deep brain stimulation? That is our fundamental, let's say, tool that we have to interface with the nervous system, is our ability to control the electric field generated by our stimulation. And then second, how do then neurons respond to that applied field? So that's really what, is the basis of brain activity, is the underlying neurons in the brain and how they are firing in response to either the electrode artificially stimulating or naturally in the brain as part of whatever system they're into. And then once we had some knowledge about that, then we wanted to actually try and implement some kind of technology that could improve clinical outcomes from deep brain stimulation. So, but before we go into all of those kinds of details, there's a really important fundamental sort of piece of knowledge that I always think is important to remind myself of as much as I can. And that is that with deep brain stimulation, or in any neuromodulation, our fundamental goal is to control the release of neurotransmitters. And so I brought up that term of neuromodulation dose a little bit ago. And I think that is a key feature. And that is that our dosage is obviously an electrical dosage, but our fundamental purpose of that is that we're trying to induce or control the firing of action potentials in neurons so that they subsequently then go and release their neurotransmitters under our volitional control. So we are using the drugs that are within the brain themselves and using them to control the networks in a way that would produce therapeutic effect. And in whatever disorder it might be, most of the time they're related to dysfunctional firing and then subsequent dysfunctional neurotransmitter release in the brain. And electrical stimulation or other forms of neuromodulation provide an opportunity for us to correct that abnormal neurotransmitter system in the brain. So with that in mind, then we can move on to trying to think about, okay, how do we build the clinical correlations of interest to us? So if we have various stimulation parameter settings in a population of patients, so this is the deep brain stimulation electrode, and you can imagine this radiating electric field coming from that deep brain stimulation electrode, then the obvious thing that we have been doing as clinicians and clinician scientists for decades now in deep brain stimulation is trying to build correlations between our stimulation parameter settings and then our clinical outcome measures, right? So if we have patients with Parkinson's disease and they have a deep brain stimulator implanted in them, we can test a variety of different stimulation parameter settings, and we can look at then how did they respond? Did their tremor get better? Did their bradykinesia get better? And we can quantify that and build statistical relationships around that. And that has been sort of the, let's say, the backbone of clinical deep brain stimulation research for the last, I don't know, let's say, 2 decades. But that's kind of unsatisfying to me because there is no underlying, let's say, scientific understanding of why those correlations exist. And so if you really wanted to get down to the meat of it, you need to understand this step of the process. So how do you go from your stimulation parameter settings to a subsequent neural response and then the subsequent control of and release of neurotransmitters within a given brain circuit to a clinical outcome measure, right? To me, that is where we want to be going, and that has been sort of the driving force for the kinds of work that I'm trying to work on. And if we then understand that system, now we have the engineering blueprint to go from a desired clinical outcome to a desired neural response to then the prospective and engineering design of the most appropriate and optimized stimulation parameter settings to actually get you to that point of being able to have the best possible outcomes. So you want to, we actually want to turn this system upside down on its head. So how are we going to do that? Well, as I said, the first step in that process is trying to understand and quantify the electric field that's generated by neuromodulation. So we can do that on many, many different levels. This is an example of a very detailed computer model of an individual patient with deep brain stimulation. So you can see this is an MRI of the patient's brain. This is the front of the head, this is the back of the head. And here's our deep brain stimulation electrode. You can see that these electrodes have 4 individual contacts on them, these little pink things. And then we can fit the neuroanatomy of that patient to the MRI so that we can have an idea of what are the different structures that surround these implanted electrodes. And so you can see here this green volume is the subthalamic nucleus. So this is the typical surgical target nucleus in the brain for the treatment of Parkinson's disease. And then we have this yellow volume here is a representation of the thalamus. So an anatomical structure right near our deep brain stimulation electrodes. And then we can use a variety of different additional imaging technologies. This particular image is what's called a diffusion tensor image. It's a special kind of MRI. And it can help you characterize what they call the anisotropy and inhomogeneity of the brain tissue around the electrode. And so what we can do from a computational perspective is to take all these different pieces of imaging data and put them into something that we call a finite element model. I'm not going to go into the math and the details. I'm sure no one could care. But I know about them and I know how to use them. And so these are computational modeling techniques that can then allow for us to solve for the voltage distribution or the electric field in time and space around our deep brain stimulation electrodes. And so this is important, as I said before, because we need to understand the spread of this voltage and the spread of this electric field so that we can then understand how it might be interacting with these anatomical structures that we are targeting from a surgical perspective. So you can do this, and these models are built off of sort of the first principles of physics. So you're basically using like Maxwell's equations, reduced down to the specifics of electrical stimulation. And these kinds of models have had amazing utility in all kinds of different fields. But no one believes a model until you actually try and validate it. And so we actually have gone through that in a very painstaking way. So this is an example of the kind of validation process that you can go through when building these kinds of models of brain stimulation. So this is an example of an experiment in a non-human primate that has a deep brain stimulation electrode. And then you can use a recording electrode to actually pinpoint various locations in the brain so that you could record what is that voltage and what is that electric field as it permeates through this deep brain stimulation electrode. And so this is almost a perfectly analogous situation to exactly what we do in humans. And so we have a situation where, but in humans you could never do these kinds of experiments because they take a very long amount of time. And so while a human is in the operating room for a few hours, that wouldn't be enough time to then be able to collect this kind of experimental data. And so we can collect this kind of experimental data so that we can actually quantify on a point-by-point basis this voltage distribution in space around our deep brain stimulation electrode. And so the reason why we would do this is because we want to test and see whether or not models can actually do this as well as or reproduce this experimental environment. Because if we could validate that the models can do this, then we can use those models in millions of different applications, but without having to sacrifice or use so many experimental resources. So, so to try and demonstrate that concept, then we built then these very detailed computer models of the individual monkeys' brains, just like we did in the human example a second ago. And then the benefit is that then this model provides us a map of the stimulation as a function, not just in these few data points, but in the whole brain in a simultaneous simulation. And if you then take those points from that model and do point-by-point comparison with the experiment, you see that you can generate unbelievably accurate and very well, very closely represent or mimic the experimental situation. So that gives us confidence then that these kinds of models are tools that we can actually use to help us understand what might be going on in the experiment or obviously in the human application. So then we're set up to try and do this process. So we want to have representation of our stimulation parameters and then make this connection between the clinical outcome measures, but really we want to make this intermediate step along the way of understanding how do neurons respond to that stimulation. So this is a pictorial example of 3 different possible, let's say, actors in this play of deep brain stimulation. So we've got all kinds of different parts of brain tissue around our electrodes. So in this picture, let me kind of back up and give you some landmarks here. So here's obviously our deep brain stimulation electrode reconstructed in a human brain. This green volume here is once again the subthalamic nucleus, so that would be the anatomical target that the surgeon is sort of using to place their deep brain stimulation electrode. And then this yellow volume is the thalamus, and then this blue volume is another nucleus nearby. It's called the globus pallidus, okay? So don't worry about the details if you don't understand or know the nomenclature. It doesn't really matter. The important point is to think that the brain has many, many different nuclei in it, and each one of those nuclei has a whole bunch of what they call cell bodies in it. So that's where the let's say the main piece of the neuron resides. And then that neuron is connected to other nuclei in the brain, and it connects itself using axons. And these axons project to other gray matter areas or nuclei, and then they make synaptic connections then with the neurons and cell bodies that are in that part of the brain. And so there are all kinds of different neural pieces, if you will, that surround these deep brain stimulation electrodes. And we don't yet have a very clear understanding of exactly which of these neural pieces are the ones that are responsible for therapeutic benefit and which ones that we might be stimulating that might be causing side effects. So in this particular example, we've got 3 different actors in our play. So one is this group of subthalamic nucleus neurons. So these are the guys that have their cell bodies and dendrites inside this green volume. And then they have axons that project across this area here called the internal capsule, and then they send their axons into the globus pallidus. Okay, so that's one group that we're thinking we're stimulating. And given the fact that subthalamic nucleus deep brain stimulation is putting the electrodes there, always the prevailing hypothesis has been that these are the neurons that we are trying to stimulate for therapeutic effect. Okay, but then there are other neurons that we are probably trying to avoid because they're causing problems if you stimulate them. And one example there is what's called the internal capsule pathway. So these are neurons that have their cell bodies actually in cortex, in motor cortex. And then they have their axons that project down through this region and then course around the subthalamic nucleus and then project down into the spinal cord and midbrain areas. And so, so these are really important neurons because they're the ones that basically take the output of the motor cortex and send it down to your spinal cord to tell you to, tell your muscles to move. But you don't want to stimulate them inadvertently, and if you do that, you can get tonic muscle contractions with your deep brain stimulation. So these are guys that we probably want to avoid, whereas these are guys that we probably want to stimulate. And then there are a whole bunch of other pathways that wrap around these deep brain stimulation electrodes that might also be either good guys or bad guys, and we don't necessarily know right now. So one example is this pathway that goes from the globus pallidus to thalamus, but in the course of its trajectory, it actually runs right by the deep brain stimulation electrode. And so this pathway is called the lenticular fasciculus, and so its cell bodies and dendrites are here in this blue area. The axons then project over into the thalamus, motor area of thalamus here, and once again, but their axons pass right by the deep brain stimulation electrode. So maybe these guys are good guys, maybe they're bad guys, we don't know. But we can use these kinds of models to help us figure that out. So how do you represent these individual neurons? Well, you can do that using a technique called multi-compartment cable modeling. And once again, I'm not going to bore you with the details of how these things are done and the math that goes behind them. But suffice to say, these are models that have been in the world of neuroscience for almost 60 years now. And they're what are called Hodgkin-Huxley style computer models. So they allow you to represent the individual sodium channels and potassium channels that are responsible for the generation of action potentials in neurons. And action potentials are the fundamental signaling unit that each neuron in your brain uses to communicate with other neurons in your brain. So we can build electrical models of those kinds of compartments of the individual neuron, representing, once again, these sodium-potassium channels. And so now if we've got these trajectories that I showed you a minute ago, and we can break those trajectories up into a whole bunch of little pieces, now we can have this electrical representation of an individual axon in this particular example. And so if we have that model of an axon, then we can couple that with our model of our deep brain stimulation electric field, and then we get then this polarization, if you will, of an individual neuron. And so now we've got all the building blocks to then be able to evaluate whether or not this particular neuron or axon will be excited by or generate action potentials in response to our applied stimulation. And so the reason I'm showing this as an example with an axon is because from the biophysics of electrical stimulation, we know that these axons actually are the most excitable things to electrical stimulation. So even if you have neurons with their cell body nearby, you're actually stimulating their axon to generate your effect with your neuromodulation technology. So, okay, now we've got this polarization of the axon. And back in the 70s, Don McNeil, who was one of the most amazing biomedical engineers ever, he figured out the math and the basic biophysics of electrical stimulation and recognized that the polarization of an axon to an external electric field is related to the second spatial derivative of the voltage distribution along that neural process. So if you don't understand what that means, it doesn't matter. Once again, I do understand what it means, and I use that technique to then allow us to create simulations of action potential generation and propagation within and around our deep brain stimulation electrodes. And so, so now we've got this polarized axon with its electric field imposed upon it. That stimulus pulse that comes out of the deep brain stimulator looks something like this. And then it causes then a depolarization or an exciting stimulus onto that axon nearby where the active electrode would be. And if the stimulus is strong enough, you will generate an action potential, and then that action potential will propagate in both directions along that neuron, and it will propagate all throughout the structure that that neuron connects to in the brain. But an interesting thing about electrical stimulation of axons and neurons is that it's a binary effect. It's either a yes or a no. It's a 1 or a 0. You either have enough stimulus to generate that action potential or you don't. And so even if you had, twice as much amplitude, you still only get one still only get one action potential. If you had half as much amplitude, you don't get a little action potential, you get nothing. So it's a binary signaling mechanism that the nervous system uses. And when we're using neuromodulation and electrical stimulation, we're also operating on that binary system. So now let's go to this big giant mess of our deep brain stimulation electrode in a human brain. And so here's our electrode. This is the contact we're going to stimulate through in red. And we've got these 3 different example populations of neurons surrounding our electrodes. So now let's try and put all these things together and see what happens. So let's put our electric field onto all of those neurons so we can see how they're polarized as a function of the specific stimulation parameter settings. And you have to keep in mind that these devices will allow you to select well over 1,000 different stimulation parameter combinations. So each one of those different combinations is going to cause a little bit different polarization and then subsequently a little bit different neural response. But in this particular example, if we then put that stimulation onto those simulated neurons, these are then the ones that get action potentials in them, right? So we have a subset of that giant population that then would be directly stimulated. And so, kind of interesting, we see that we get lots of these subthalamic nucleus neurons just like we would have expected because that was the underlying hypothesis. But you actually get just as many, if not more, of these globus pallidus axons that, interestingly, you can also stimulate in this blue volume with your deep brain stimulation electrode and get a good therapeutic effect. So that it's not just one target that we're shooting for. There are many, many different neurons around our electrodes, and it's quite likely that we're stimulating lots of these different pathways when we're getting our therapeutic effects. And it's actually that many of these pathways would be important for us getting therapeutic effect. So, so then we're faced with a complicated situation because the kind of model that I just showed you is interesting and potentially informative from a scientific perspective, but it is totally unwielding and not, it requires basically gigantic supercomputers working nonstop and a team of graduate students to generate those kinds of results. Right? So that is completely useless in the context of finding a way to get that knowledge into clinical applications. Because clinicians don't have time or expertise to be able to manage big giant computer models like that, nor do they have interest in it, right? To them, this is a tool, it works to help our patients, and I want to be able to adjust the stimulation quickly and easily. And I don't care whether it's this pathway or that pathway or whatever. I just want it to work. So with that in mind, Chris Butson and I worked for many years to develop a simplified version of that kind of simulation, with the idea being that if we could provide that kind of visual feedback of stimulation parameter adjustment to the clinician, they would then have a tool that could help them along the way. And so that concept, what came out of that concept was this idea of a volume of tissue activated. So basically this is a simplified version of the kinds of models I walked you through that probably has like 90% of the backbone of that very detailed model, but it can run on effectively an iPad. And so that you have this much more realistic clinical tool that could be put into the hands of clinicians. And so once again, you've got this anatomical model of the patient's brain, an electrical model of the tissue medium, you've got your stimulation parameter settings, then you calculate and solve for a voltage distribution, which is then coupled to predictions of axonal stimulation, and that can then be summarized into a red activation volume, if you will, that would be adjusted by or dictated by the specific stimulation parameter settings that you would be using in that particular patient's brain. Okay, so once again, don't worry about the details if you don't really want to know, but the concept being that now we have a tool that we could put into the hands of clinical visualization technologies. And so, so we have an academic version of this software. There's also a commercial version now that's owned by Boston Scientific, and it's available in Europe. It's called Guide DBS. So they have taken this technology and actually commercialized it into clinical practice. But in this particular picture, once again, we're looking at the surgical targeting knowledge and information that would be available to the neurosurgeon to plan where to put the electrode. Then they would do this intraoperative electrophysiology, once again, probably using FHC electrodes to do that. And then we've got our implanted deep brain stimulation electrode relative to these points of electrophysiology, as well as the anatomical structures. And now we can then overlay that with our deep brain stimulation activation volumes that would then be relative to or dictated by our stimulation parameter settings. So now we've got a tool that allows us to actually make these kinds of clinical correlations. So we want to go from our stimulation parameter settings to clinical outcome measures and understand the neural response. And so Chris Butson went on to then push that concept by evaluating stimulation in many, many patients, so over 100 different stimulation conditions tested, to basically map out this subthalamic nucleus region of the brain. And in each one of those little red volumes, there are many of them overlaid here, you could quantify a specific amount of improvement in bradykinesia or a specific amount of improvement in rigidity. And if you then had this map of stimulation, if you will, across many patients and across many testing situations, you could build what is called a probabilistic stimulation atlas. The concept being that it would allow you to hone in on the target area that if you stimulated these parts of the brain, you would get the best improvement in bradykinesia or rigidity. And so that we use the model to back calculate where is it that you're going to get the best possible clinical outcomes. And if you have that information, now once again, you're ready to make this step from here is our desired clinical outcome effect. We can quantify what that neural response needs to be to get to that effect. And then from that, we can then optimize the specific selection of the parameter settings so that we get to that quickly and easily for the neurologist. So we put this concept into prospective testing. And so the goal of this particular project was that we'd have our target volume that was derived based on those probabilistic stimulation maps. And we'd say that, based on all of that data that we had collected on patients from the past, we know that if we concentrate our stimulation in this area, we'll get the best effects that we can. And so then we can use the model to calculate this volume overlap and pick a parameter setting that would be theoretically the best we could do. And then we could do a head-to-head comparison between the stimulation parameter settings that were defined based on traditional clinical practice. So this is the original slide that I showed you at the very beginning, where the clinician would go through and test many, many different settings until they found the perfect one for that patient. But once again, that was a very time-consuming and difficult process. It's very much dependent on the expertise of the clinician. Whereas this is something that would be an automated computer algorithm that would be using science and anatomy to come up with that predicted optimum. Okay, so when we do the head-to-head comparisons between our model DBS, or our model DBS and the clinical DBS, we saw that we could generate equivalent improvements in therapeutic benefit using this approach. And the idea being that this is the goal that could then be the start point for the clinician to interact with the patient and then refine that selection of the optimal parameter setting. But one of the interesting things that came out of this was that not only did the model setting do well in improving the traditional Parkinson's rating scale, it also actually alleviated a common problem with deep brain stimulation, and that is that there can sometimes be some mild cognitive side effects of the stimulation. And so we hypothesized that that was related to an unintentional overstimulation of the area around the STN. So if the motor part is in this area, if you turn the stimulus up too much, you could get into other circuits in the brain that are things that you don't want to be interfacing with. And if you could have that perfect balance of just motor stimulation, you would maintain the ability to do well on this task, which is a cognitive motor task. And if someone's really interested, I can give you more details on how you quantify that, but I'm not going to get into it now. And then the next generation of technology is going to then be taking that and pushing it even further. So the next big concept that will be available in commercial devices, it's already available in Europe, hopefully soon it'll be available in the United States, is then an idea that we could use multiple contacts to steer the current through the stimulation so that we focus the stimulation in a much more refined way. And so I'm going to show you a little movie that does that. So what we're looking at here is now taking this stimulation volume, 2.5 milliamps, and then shifting it slightly in increments from one contact to the next contact to the next contact. And then if you had this visual interface that allowed you to see how that stimulation was adjusting, you could then be able to refine it so that it would interact with and find that happy medium, if you will, between your target and the capabilities of your stimulation device. And so, as you can imagine, as these devices become more and more complicated, we have more and more electrodes and more adjustment opportunities within those electrodes, you're going to have basically this infinite parameter space that the neurologist is going to have to navigate through. And they're going to almost have to rely on these kinds of engineering technologies to help them identify then the best possible stimulation parameter settings. So, I guess in trying to kind of come to a conclusion and summarize some of this stuff up, I think that an important concept as we move forward in the development of neuromodulation technology is that we have to start thinking about what is our neuromodulation dose. And so remember that the fundamental point of what we're trying to do is control the release of neurotransmitters in the brain. And that is what we're doing via the generation and control of action potentials, given where we place our electrodes. And that's then dependent on the electrode location, the stimulus pulse, and the stimulus frequency that we're actually using in these applications. And then while we would like for things to be selectively stimulating only parts of the brain that we want, the reality of the situation is very different than that. And that is that we're stimulating many, many different pathways in the brain. And these pathways are the connections between various brain networks. We're only now just at the very beginning stages of even understanding what these networks look like and how they're connected to each other. But this picture tries to give you a sense of what some of these possible networks are in the context of Parkinson's disease. So here we've got our cortex connected to these deep brain structures. So we spent a lot of time today talking about the STN, and then how it's connected with the globus pallidus, and then how that's connected with thalamus, and then that comes back up and connects with cortex, which comes back down around, and it creates these oscillatory loops, if you will. But there are lots of different oscillatory loops within this system. So there are little loops, like this loop between the subthalamic nucleus and the globus pallidus, and there are big loops that kind of go all the way around the wheel. But these guys are interconnected with each other, right? So the STN's in this loop, and it's in this loop, and so how do these things interact with each other? Well, I like to think about it like this kind of old clock analogy. So we've got these little cranks, we've got big cranks. You turn one, you're going to get an effect on the other. We don't necessarily know exactly what those effects are going to be, but we definitely know that they're turning each other in ways that are very innovative and interesting. And if we start to understand the dynamics of those interactions, now we've got a whole new way of trying to communicate with the nervous system. And we can do that with our neuromodulation technologies, right? So we can come up with all kinds of interesting stimulation paradigms so that we're driving these gears in exactly the right way, and thereby improving our outcomes and minimizing our side effects. And that's really the new frontier in this field. So I guess I'd like to say thank you for listening, and hopefully you got something out of this. And I'd like to thank the National Institutes of Health that paid for all of this. So thank you.